Do you want me to? Okay.
Yeah, you got it.
All right, we are ready? Okay, everybody, is this working all right? Can everybody hear me okay? Thank you so much for coming to join us. This is our first Science Cafe in the Bangs Community Center. And Science Cafe is now coming through Science Stories, which is, if you don't know, a graduate student science communication organization run by us grad students at the University of Massachusetts Amherst. So in the past, Science Cafes have been running since 2011, bringing science to the Valley. In the past, we were under the organization name Life Science Cafe. We've merged with the That's Life Science blog to create one sort of big umbrella science communication organization. So if you don't know us, please check out our website and our social media pages. Our website is Science Stories. It's on some of our flyers. I'll get you the website after the event. Don't have it off the top of my head. But we're so grateful for all of you for coming out. This is our first Science Cafe back out in the community in person after our hiatus. So thanks for turning up. And we are excited to be here to talk about science with Dr. John Gibbons, who I'm going to introduce now. I'm Thomas. I'm a PhD student at UMass. And John here, we're so excited to welcome him to talk with us here tonight. Dr. Gibbons is a professor in the food science department at UMass Amherst. And he studies microbial domestication and population genetics, which we are about to hear all about. John earned his bachelor's in science and biology from Keene State College in New Hampshire, his PhD in biological sciences from Vanderbilt University, where he worked in the lab of Antonis Rokas. I'm not sure if I'm pronouncing that right.
Pretty good.
Okay. Dr. Gibbons was also a postdoc at the Harvard T.H. Chan School of Public Health, and then a faculty member at the biology department at Clark University for 4 years, before moving to his current position as a scientist here in our community. So please help me welcome Dr. Gibbons. Thanks. So, John, we are here to mostly talk about your research in food science, but before we get into the technical stuff, we want to know a little bit about you. So can you tell us about your introduction to science? What kind of got you interested, or how did you come to a career in research?
Sure, yeah. I wish I had a nice backstory, but I kind of floundered a little bit in high school, and I wouldn't say I ever really was super drawn to science. And I think it was college, so I played soccer my whole life, and I really liked filmmaking. I used to make stop-action movies when I was younger, and I chose Keene because I thought I could play soccer there, and they had a film major. It was the only public school, I think, in New England at the time that had a film major. And then I broke my ankle, which was actually like a blessing in disguise, and I got really serious about school, and I took a non-major course on speciation, and it like blew my mind, and I decided to major in biology after starting in film and going to psychology and then sociology, and I finally landed on biology and kind of went from there.
Very cool. So are you back to playing soccer at all?
I am, yeah, but also breaking my ankle and spraining my ankle all the time. But
yeah, oh no, for sure.
Yeah.
No, that's great to hear. I was kind of a high school flounderer, so I always love hearing that from faculty and researchers. So speaking of your hobbies, you mentioned soccer, stop motion, filmmaking, and before we get into science, I had to ask, because you mentioned this in your email to us, that you do metal detecting or metal detecting?
Yeah, I feel like very old talking about it. It's like a very old person hobby, I think. But so there are these really cool old abandoned towns all over New England, and I go with my metal detector and try to find old coins and artifacts and stuff like that.
What's the coolest thing you've ever found?
Oh, that's kind of depressing. I don't find much, but you find a lot of bottle tops, and but I found like some buffalo nickels before, and some really intricate padlock from like the turn of the century. There's a place called Catamount in Massachusetts, pretty close by, and it was a town at one point, and it's a state park now, and if you hike it, you're like, how did anybody live here and farm here? It's just all hills and rocks. So they got out after, by the early 1900s they were gone, but you can find artifacts all over. Yeah.
Wow, that's really cool. So biology, you told us a little bit about how you took this non-major speciation course that kind of brought you to biology. So why microbes? I mean, so you study food science, microbes. What was the sort of connection point for you there from the speciation course into food science? And
I don't think I've ever been drawn to an organism. For me, it was always the questions, and so in grad school, I was really lucky to get a great advisor I was his first PhD student, and he studied fungi. So I was like, all right, I'll study fungi. Why not? So he gave me a bunch of papers the first week, and he said, what's interesting? And read these, and let's talk next week. And what's interesting? And I found some papers that I thought were really cool, and we designed some projects off of that. But since through my postdoc and even after, I've worked on fungi, I've worked on humans. We had a polar bear genomics paper. I've been involved in viral papers, and so for me, it's always kind of been the question.
so what are some of the questions that you're seeking to answer with your work these days?
Yeah, thank you. That's a big picture question. This whole idea of domestication to me is just like so fascinating. How humans could shape living things just by breeding them. It's really wild. And so, for me, there's the historical aspect of it is so interesting. It's like you're looking back in time and trying to figure out why did people do the things that they did. And so, humans have been around anatomically modern humans for 200,000 years or so. We spend all of our, most of our history in Africa and successfully leave 60,000 years ago. We breed with Neanderthals, and all this stuff happens. And it's only the last 10,000 years that we've learned how to farm and stopped, following food around. And so that had always been really exciting to me. And then, you hear about dog domestication and, maize domestication, but there are these microbes behind that are also, that were domesticated by humans. And it's something that isn't talked about much, but was so interesting to me.
So a lot of the time when those of us who aren't in this field are thinking about molds or fungi or bacteria, we're kind of thinking about pathogens, sickness, or issues for human health. But there are some positive or sort of beneficial relationships you're saying that we have had historically and presently with some of these organisms. Can you tell us a little bit about some of the areas that we may not be thinking about?
Yeah.
Or microbes at work?
Sure. I mean, imagine our whole history we're trying to find food and traveling seasonally to get food and to track animals, and finally we're sedentary, and there's an abundance of food because we're farming. But how do you preserve that food, right? Now this is the first time there's an abundance of food, but it can spoil, and so we think that most of the organisms that are used, microbes that are used in food production, are there because they've limited spoilage, right? They're increasing the length that you can eat food. And, I mean, you think about raw milk will spoil in hours, and if you ferment it into a hard cheese, you can travel around with it at room temperature, for months. It's really wild. So we're trying to figure out, what was the series of events that happened with microbes, and how did humans change their traits, and how did they change their genomes?
Very cool. So when I think about domestication in that sort of sense of changing genomes, I'm often thinking about dogs, cats, corn.
Yeah.
So how would you define domestication, and then how do you domesticate something so small as a yeast or a microbe?
Yeah, that's like the big question. How do we do that? So I think domestication, you can define as humans breeding something in isolation from its ancestor, and selecting for traits that are beneficial. So, if we think about dogs, you have this huge variety of breeds, and they're all they're all bred for a certain trait. You know, it could be hunting or tracking, things like that. For microbes, and they look very different from their ancestor, right? With microbes, you look at them and they look the same. The wild ones, the ones that are used in food. So the idea this running idea is there's this process called backslopping. And so you can imagine a long time ago you have this really good fermented thing. It could be like sake, rice wine, okay? And so it turned out really good. And so you scoop out a little bit of it and you put it in the new batch that you're going to ferment, hoping that it creates the same type of fermentation or the same product. And so the idea is that you keep doing that and you keep doing that and you keep doing that. These microbes are basically getting serial transferred in the same environment generation after generation after generation. And they start adapting to the food that they're grown in.
So this is, I mean, kind of similar to the process you're describing to keeping a sourdough starter,
Yeah.
making yogurt at home.
Exactly.
We're engaging in that same process.
Yeah.
So can you tell us a little bit about, that's the sort of big picture of the questions you're asking. Can you tell us a little bit about what your day-to-day research looks like? So like, are you in the lab making these sort of mediums for microbes?
Yeah.
Are you sequencing? I mean, what does it look like?
Yeah, so we have two models that we've developed a bit. One is Aspergillus oryzae and Aspergillus flavus. So Aspergillus oryzae is a domesticated mold that's been used for thousands of years. It's used to ferment rice products and soy products. So sake, soy sauce, miso, these are all products of Aspergillus oryzae. We know in the wild there's Aspergillus flavus. And if you look at their genomes, they are 99.5% identical. I mean, it's like if you look at the person next to you, you're sharing basically the same genetics, you know, the amount of difference. Super, super similar. So we have been studying their genomics and their evolution of Aspergillus oryzae and flavus. I started this in grad school, and we're still working on it. And we get as many strains as we possibly can, and we sequence their genomes, and we use those sequences to try to infer their evolution.
So to sequence the genome, give us like an image, sort of. If I walked in where you're at work, are you at a computer? Are you sort of sitting down at a machine?
Yeah, unfortunately, the people here do most of the work. So we have some grad students here, Adenike and Nadia and Ezra as an undergrad, that all work in our lab, and they do most of the lab work. I love going up into the lab and bugging them when they're doing stuff, but. So basically, you know, we are growing these molds they grow on anything. If you give them water and a carbon source, they will grow. They're generalists. And so we have some standard media that we grow them in, and then you're basically taking some of that fungal tissue, grinding it up, and trying to extract DNA from that. So with that DNA, you send it off to, you sequence its genome, we send it off to a company for sequencing, and then we get an email. It's always a happy day when it comes in, and then we get to start processing it. And UMass has this great, it's a shared supercomputing cluster that we do all of our analysis on. Okay, so that's usually like, you know, I will get in that stage, or even a little later with the analysis.
Yeah, some of the big data sets. So I want to talk a little bit more about some of this recent work that you've been doing with genomics, but I also want to pause. So we're going to take a couple breaks during our conversation with John, so that as questions come up for you all in the audience or on Zoom, you get the chance to sort of ask him things before we move on to other topics. So I'll pause here for a minute, and if anybody has a question, or if anybody in the Zoom room has a question, you can raise your hand or your Zoom hand. Yeah, do you want to come up to the microphone, or I can try to bring it to you? Thanks, that way we'll catch you online.
Thank you. Thanks. John, I was wondering if any of your Aspergillus lines have become pathogens or degraders, or have you seen that at any point in the sort of co-domestication?
Yeah, that's a good question. So Aspergillus is this really big genus of fungi. There's 300 species or so, and we call them my advisor, Dr. Rokas, used to call them the Dr. Jekyll and Mr. Hyde of fungi, because you have pathogens that cause lung infections and kill hundreds of thousands of people a year, and you have these other species that are used in food fermentation and to make drugs and to make citric acid, and we use them quite a bit. So there are cases of people getting a fungal infection that work in that brew sake because they're in constant exposure to these spores. Typically, they're not pathogenic. Aspergillus flavus, the wild one, is rarely pathogenic. It can happen, but there is a species called Aspergillus fumigatus that is like a super infection agent for people that are immunocompromised. We have some experiments that we're trying to go to the opposite direction. So we're starting with the wild one, and we're saying, all right, if we keep growing it on rice, and keep growing it on rice, and keep growing it on rice, can we get it to look like the domesticated one? Like, can we mimic domestication in the lab? And that's work that is kind of still in progress.
I'm glad you're going in that direction. The other one is kind of like mad science, evil genius, right? So you have this recent paper where you are looking. Oh yeah, sure. Sorry.
I have a question. You are in the food science department. Do you consider yourself more of a food scientist or more of an evolutionary biologist, and what do you think the difference would be?
Yeah, I am definitely not a food scientist, but we do a lot of research that is food-based. So I'm definitely, I was trained as an evolutionary biologist and as a genomicist, and I've loved getting into the food science department. So we work on these food microbes. We also work on other types of fermented foods that are spontaneously fermented, and we work on bacterial pathogen, the foodborne pathogen Listeria. So we work on other food systems, but for me there's so much application. It's so much more applied, and I found that like super interesting and also very very challenging at times too.
It's very present in our lives.
Yeah.
What is spontaneously fermented?
Yeah, great question. So cocoa beans are an example. Cocoa fermentation. So when chocolate is actually fermented, it's in these big pods. Open the pods, and these white fruits that are they're just basically heaped, and whatever is in nature is fermenting them. So this could be from like insects flying in that have microbes on them and crawling around. It could be whatever is native to the outside of the pod. It's environment, dust that's rolling around and lands on it, that's in the wind. These are spontaneously fermented from things that are in nature. Yeah. Got it.
So really just kind of picking up on wild microbes.
But very reproducible, which is really interesting. Yeah.
So are there any other questions at this point? Or we can keep going. We'll have more breaks for questions. Did you have a question? So I want to ask you about this aspergillus research you've been doing. So you were looking at these aspergillus strains that are used in sake fermentation in this recent paper that's just come out of your lab, and looking at populations that are used in food production, comparing them to their wild ancestors. Mm-hmm. So first question, can you make sake from both of these organisms? And if so, are the domesticated ones better?
Oh yeah, cool, really cool question. So you wouldn't want to do it from the wild ones because they produce, many of the strains in the wild produce toxins, and some of them are super potent. So you've probably heard not to eat moldy peanut butter. It's because it can be Aspergillus flavus or Aspergillus parasiticus, which produce aflatoxin, which is the most carcinogenic compound that we know of that's naturally occurring, which is wild, right? That a fungus produces this. And so, like in the U.S., it's federally regulated because it can contaminate crops, stored beans, like dried in silos. If it's too moist, you can get fungal growth. It's regulated in milk, so because of feed. So it's you'd never want to have the wild ferment your sake.
So the domesticated sake, definitely.
Yeah, but thanks for bringing this question up. It's what we actually haven't published this yet, but Colin McCarthy, who's a master's student in the lab, has done just that. He's basically fermented rice with some of our wild ones and some of our domesticated ones, and then we do something called GCMS, where we're trying to pick up on the volatile compounds that are present in our samples. So while it's fermenting, what kind of odors and aromas is it giving off? And in some of the literature, they say that the wild ones give off like more of a like a musty, earthy smell, and the domesticated ones give off what someone has described as like this sweet grapefruit smell, a little bit cheesy too, which kind of doesn't sound great for sake. But so we did this analysis and we figured out, we identified which volatile compounds were present in the samples, and what was amazing is the domesticated ones actually produce like 25 or 30 more compounds than the wild ones. And these are, many of them, or most of them, were associated with like these very pleasant floral odors.
Wow.
It was really cool.
But a taste test off the table.
Probably with the wild ones, yeah.
Yeah.
But I used to be a hobby before I had kids, yeah, was home brewing sake. Yeah.
Very cool, yeah. So you've found some of these differences recently in these volatile compounds. With the population genomics work you're doing comparing these lineages, what kind of difference did you find? And
Yeah, so the work I think that you're talking about is actually with a different pair of species.
Okay.
That Kim Acevedo, who's a PhD student in OEB, has been working on for her thesis work, and has done some like really exciting work. So Soji is the domesticated one, Parasiticus is the wild one. And Soji is really used for soy fermentation, not for rice fermentation. And so she sequenced the genomes of 20 of these, and then there's all this public data. So we've analyzed maybe 40 strains of the two different species. And when we look at how they're related to each other, what comes out is that there are 4 major groups of the wild one, which is consistent. Like you have different populations in nature. And the domesticated one is a very tight population with almost no genetic diversity. And so what that's telling us is that, whatever strain was used initially was then, was basically clonally evolved. And so it's used in all these other, we have them from Korea, from Japan, from China, and they're all pretty much the same genetically. So it was kind of a little bit surprising because the other comparison we look at, the Oryzi flavus comparison, is a little different. Yeah.
so that's kind of a strong indication that it's this really maybe specifically domesticated or cultivated population.
Yeah.
Kind of
Yeah.
from is that indicating that it's from a single source lineage?
Yeah,
Or?
that's one of the cool parts. So there's one of these wild populations is most closely related to the domesticated population. And when you look at their genetics, the wild ones, you see both signatures of the wild genetics and some of the domesticated genetics, which means that much of the genetic variation that you see in Soji was already existed in nature, and we call it like it was standing genetic variation that already existed. Yeah.
So sometimes we think of reduced genetic diversity in a population maybe as being a bad thing, or for a food system, a point of vulnerability. Is this sort of, in this domesticated population where you're seeing kind of less genetic variation than the wild type, is that kind of comparable to what we see in maybe other like domesticated food crops or other domesticated species and other types of
Yeah, it's a great
animals?
question. One of the big like signatures of domestication in plants and animals is seeing like a big reduction in genetic variation in the domesticated populations, and the same is true for fungi. So with our examples with Aspergillus soji and Aspergillus oryzae, there are some groups in France that look at the penicillium fungi that are used to make like camembert cheese and blue cheese. And they've actually said the genetic variation is so low in the blue cheese fungi, that we have to think about conserving it. How do we conserve these? The caves are getting warmer because of climate change, and they're trying to figure out ways to actually preserve the fungi.
Wow, that's an interesting question. And I was going to come to this later, but maybe that's a good time to ask this question. So since you're working on these organisms that have these significance for food systems and kind of these histories of domestication and cultivation, what are the kind of cultural impacts of your work? Is this shaping how people are thinking about these historical traditions, thinking about carrying these food ways into the future with changing climate and that sort of thing?
Yeah, that's a good question. I don't think our work is that exciting. But I do think it gives you like a window into the past. And I think that is worth a lot. And when you think about the history of fermented foods, I mean, they're like sacred in human culture, especially the ones that have alcohol. So like sake is used, is in traditional Japanese religion, Shinto. It is sacred, sake. The wine is quite literally in the Catholic Church, like, the blood of Christ, and the pulque, which is a traditionally fermented Mexican drink from agave sap, was sacred, and there's Mayan paintings that depict it. It's so, culturally fermented foods are really important, and I think that's what I always kind of come back to.
So to kind of get to that historical perspective that you started out with, can you imagine, this is kind of speculative, but if we had never domesticated these microbes or kind of entered into this relationship of domestication, I don't want to like attribute it all to humans. Who knows, maybe the fungi have some agency in this process. So maybe you could speak to that. But if these organisms had never been domesticated, how do you imagine our world might look different today? I mean, no sake?
No sake.
So that's a bummer.
Yeah, that is a bummer. Yeah, I mean, they're such an intricate part of our lives. So many food systems use microbes, and it's weird. They're popular now, like fermented foods, right? People like kombucha, and you can find it everywhere. And I don't think we don't have life. We don't have food without microbes. So much of traditional food is fermented. Traditional pickling and.
Yeah, so when we think about like the contemporary context of that fermentation, I know for some, I've done some home brewing, I've baked sourdough and that sort of thing, and I get yeast like online and it comes in a little packet and I keep it in my freezer, and I have no idea, and I've wondered with baking yeast or brewing yeast, where is that coming from? So for these domesticated, any of these sort of organisms that you've touched on that are used in food production where they're being domesticated, are there farms or factories, or where is that being grown, and what is that process like?
That's a really good question. So the process of maybe I could back up and talk about. So the way that the sake is fermented is you steam your rice and you put it on a very flat surface and sort of cool it down, and then you're adding this mixture called koji, which is it's wood ash, and it's spores, and it's dried rice. And that is what's actually like sort of sprinkled onto the rice to start fermenting it. So that can be dried, that's grown for a few days, and that can be dried and used again as your new batch of koji.
Got it. So kind of making a starter, you take a little bit of the previous batch.
But the nice thing about molds is that you can they are super resistant and resilient, and once you have spores, you can keep them almost indefinitely.
Right. Dry them out and wake them up again.
Exactly.
Now's going to be a good time maybe for any other questions from the audience or from the Zoom room. Take another little question break. Yeah, Elise. Yeah.
I have a question. So in thinking about what you just kind of talked about with the whole sprinkling out your inoculum onto the rice, how stable or like how much fidelity do you have in any given thing? Like if you think about like Richard Lenski's evolution experiments, those microbes are just like splitting all the time.
Yeah.
How phenotypically stable are these microbes?
Yeah. Well, they haven't started metabolizing new stuff like the Lenski experiment, which is, I guess that's good. They're pretty stable, but like with Oryzae especially, you have strains that are specialized for different things. So for different sensory attributes, for different protease activity or amylase activity, things like that. Actually, that's one of the coolest stories with Aspergillus oryzae and Aspergillus flavus. So when you're eating food and you're chewing food, in your saliva you have the salivary amylase enzyme that's being produced, helping you break down carbohydrates. And in human evolution, that gene has actually duplicated in the genome in populations that have starch-rich diets, which is really wild. The same thing has happened in Aspergillus oryzae. So the wild ones have a single copy of this alpha amylase gene, and the domesticated ones have 2, 3, 4, sometimes 5 copies of the same gene, which is really amazing. And some of our work from a couple of years ago with Catherine Chacon Vargas, who was a PhD student here in MCB, she actually discovered that they have evolved completely differently. So there are copies that are on chromosomes that we wouldn't expect them to be on. They're kind of hopping around everywhere. And the more of that gene you have, typically it correlates really strongly with the amount of amylase that you're producing. So they are evolving, they are actively evolving. Yeah.
So I'm going to cut in because I have a kind of a related question, which is I know that so I work with plants, but I have a little bit of background with agricultural mammals as well, and I know in some cases of domestication, when we look at these histories, we see like domestication syndrome, where certain traits kind of come along with domestication by accident in some ways. So like coat color patterns or floppy ears in mammals. There's sort of a set of traits in plants. Are there, across maybe some of the different species that you've worked with, domesticated species, obviously not floppy ears, but some other kind of patterns of consistent changes in some phenotypic traits like that?
Yeah, that's how we got a grant for this, is trying to say, is there like a fungal domestication syndrome that we can define? And it's tricky because there's exceptions for everything. But in fungi, it looks like, in filamentous fungi at least, it looks like there's a loss of secondary metabolism. That's one of the big domestication syndromes. So secondary metabolites in fungi are these compounds that many times are defense chemicals. So they're used to, imagine fungi are weird and they digest their food externally, and then they try to like reabsorb the nutrients. And so when the nutrients are out in the soil, their competitors also want it because it's like very easy nutrients, right? So they've evolved this amazing array of arms of different toxins and secondary metabolites. And actually, aflatoxin is one of those that we talked about before. And when you look at the domesticated ones, it looks like for the most part, they've lost a lot of that ability to produce some of these toxic secondary metabolites or these toxins. And there's a few different explanations or hypotheses. One is that you don't need them anymore. You don't have any competitors in the food environment. You have all the food you could ever want, and it's stable, and there aren't competitors. Mm-hmm. And so you're wasting energy producing these. And these compounds take a lot of energy to produce. So you're freeing up energy now from this making toxins to actually eat for primary metabolism. And so that's one of the big ideas of, this is probably the biggest characteristic of fungal domestication.
Great, yeah, that makes sense. I mean, that sort of sounds like these mutualistic relationships we have with other species that they don't need to hunt anymore,
right.
or whatever it is. Yeah, spend so much time foraging. Great. Any other questions before we? Yeah.
All right, so go co-ops. We love co-ops, but also it's kind of oversimplification to think that we can have local ag and local meat production in every town and every city. And so, I think from a public health lens, you have to consider that there's going to be really large industrial suppliers of meat, of ag, mm-hmm. and then as you think about macroeconomics with that, you're going to increase your supply chains by having single distribution points, but with that, boar's head, listeria outbreaks, etc. What advice would you give to major industrial suppliers of these products so that you can continue to do this cheaply and make sure people can afford them, but also prevent public health dynamics or adverse public health
Yeah,
dynamics.
oof, I don't know if I should be answering this. Yeah, or I'm gonna get sued or something. But
We'll
Yeah.
redact it. That company name mentioned.
it's yeah. I mean, having large-scale food processing facilities is really scary, right? Because if you introduce, a pathogen could be introduced at almost any point, and you're in trouble, and it could just sort of replicate. So really, we know what practices to sanitize surfaces and things like that. It's just harder to do at a big scale. I don't know if I should be giving advice on that, since, but really good sanitization is probably the best that we can do.
Other questions from the audience right now? And a couple more. Well, maybe following off of that, in terms of public health or consumer safety, there's the side of it that you just mentioned with the sort of recent scary listeria outbreaks, but then also, like you said, fermentation is really hot right now. You get kombucha, everybody started making sourdough, and there's all kinds of also information out there about prebiotics and probiotics and all kinds of, I don't even know, all kinds of other things. So it feels like a sort of a complicated information landscape, on this complicated topic that I don't know much about. So what do you think that those of us who aren't scientists in this field should sort of be thinking about in terms of taking care of our bodies, our food, but also finding information and kind of, where should we turn to learn what we need to know about these topics?
These are the hardest questions. I wasn't ready for these. I mean, diet-wise, study again have basically shown that the more fiber you eat, there is no upper limit. The more fiber you eat, the healthier you are, and part of that is because you're feeding your microbes, right? Fiber has all these really complex carbohydrates that your body can't digest, that microbes that are feeding your microbes. And there's so many benefits to having a healthy gut microbiome in terms of inflammation, anti-inflammation and things like that. My partner had a product recently, and I looked at the label and I was like, hey, it says Aspergillus oryzae on there, and there's they were using alpha amylase, this enzyme in this product, as as a probiotic or a prebiotic. So the FDA isn't involved in a lot of the probiotic research, which which is we don't have this governing body that looks at this stuff. So it is hard to get good information. And I'm not sure I want to lead anyone down the path to a certain source. Yeah.
Sure. Well,
Yeah.
now that I've asked you, maybe erroneously, to give advice to anybody who eats food, let me zoom around a little bit. Does working in this field generally, in any way, sort of, has that changed how you cook or how you shop or how you eat? Are there things that you will eat that other people won't, or
Yeah.
things that you won't eat that other people will?
That's a good question. I'm pretty adventurous eating, I think, but we have kids who are kind of picky, so we cook the same kind of stuff every week. Yeah, let me. I'm trying to think of. I just lost my train of thought. Sorry, what was the big picture question?
Just sort of has it changed how you,
Oh yeah.
how you shop?
So yeah,
or,
thank you. So I when I gave my teaching talk at UMass, it was on the gut microbiome and diet and how they influence each other, and that actually, just by creating that lecture and really thinking about it, did change the way that I eat. And I am mostly plant-based now, and it's really changed. It has changed the way that I eat. Yeah. I don't think I thought about it as much before I got into this department. Yeah.
Do you have a particular favorite fermented food or drink?
I do love sake. I have a special place in my heart for sake. And there's a sake brewery in Massachusetts now, near Boston, called Farthest East. and that makes unbelievable sake. It's one of the only sake breweries, I think, in New England, and there's only a few in the U.S.
Wow,
Yeah.
thanks for the recommendation.
Yeah.
I know, which all this sake talk is making me wish we could have
Yeah.
sake here tonight, do a little taste test, but we have to get all this approved through the UMass finance office, so you wouldn't believe what that process is like. I think it would be a no-go. Okay, so I want to ask you a little bit, we kind of have talked about your research work, but you're an educator too, you know, and I'm so glad some of your students are here. Can you talk a little bit about how you see your role as an educator?
Yeah, sure. I mean, I try to. For me, the most exciting thing about teaching is being, is helping people find what makes them curious. You know, what actually makes you curious? What are you driven to think about? And so in all my classes, that's kind of what I strive for. Like, what are you passionate about? And maybe you don't know what it is, and that's part of the fun. So. There are some courses that I teach that are more, that are kind of more applied in terms of doing analysis, and I teach a graduate genomics course. It's called Food Science for Genomics, but I've had students from all different departments take it, and many of them don't have any experience doing genomics work, and so part of the project, part of the class is actually doing your own original projects that you have to take ownership of, and it's amazing because some people end up using this in their PhD work after, or get a job in the bioinformatics field or genomics field and figured out that they actually really like it, and so it's like empowering people to find what makes you curious, and giving people new tools.
That's great. So you're also, in terms of that sort of, support of your students' curiosity, you're also the chair of your department's workplace climate committee.
Yeah.
Can you say a little bit about what you're hoping to do in that role, or sort of what you do to build community?
Yeah. Over the last few years, so one thing we did last year was we had a graduate student town hall. We have so many graduate students in food science. I think there's over 60, might be 70 or 80, and our building is very tight and old, and it was just basically, we held this town hall basically as a forum to ask graduate students, like, what is going well or what's not going well, what do you like about our department, what don't you like? And the idea is to get confidential feedback back, honest feedback, and see what we can do better as faculty. I'm really looking forward to presenting that to our department to say, here's what we need to do better, here's what we need to improve on, and here's what's going well.
Yeah, that's great to hear about. So you're kind of the results of that pending, you're presenting that to the department.
Actually, we're waiting for the grad student organization in food science to approve of our notes so that we can then have the discussion.
That's the one.
Yeah.
Cool.
Yeah.
Yeah, that's great to hear about. I want to pause for, I've got one or two more questions, but if anybody else, we're getting close to the end of our hour here, so as there's other things that come up on anything we've covered. Anything in the chat, Matt? Yeah, please.
Hi, John. With respect to mentorship, what advice would you have to graduate students who are looking to pursue research?
Yeah, that's a really good question.
Or are in grad school right now?
Sure. So finding the right fit, finding the right advisor. And I know that I'm probably not the right advisor for certain students, really finding the right fit. Some people like to be micromanaged and want to have more structure, and some people want to have, be kind of free to do their own thing. So I think it's so important to find the right advisor. You could love the project but have a really bad relationship with an advisor if you don't align well. So I always encourage, whenever someone's applying for our lab, and I really encourage them to reach out to every graduate student every undergraduate in our lab and talk to them about how their interactions and their relationship with me is, because it might not fit for you. And you know, if you're going to graduate school, you're going to be working on something for 2, 3, 4, 5, 6 years, and so you want to really love what you're doing, or it's going to make that period really tough. Yeah.
Other questions in the audience? Maybe just, I've like 2 more questions as we kind of close ourselves out, and then if any folks want to chat in the last few minutes, we can.
Sure.
Sticking on the topic of sort of mentorship and teaching, when you think, you know, over your career so far, are there any lessons, big or small, that really stand out that you've learned from your students?
Yeah, I mean, I know when to say I don't know. And I think that's such a valuable lesson. We're not supposed to know everything. So I love getting stumped, and it happens often. But then, you know, it's a chance to really think about things. And to be honest, I think, you know, the way that we think is more important than what we actually know. So a lot of times it's just thinking about things deeper. And you know, for me, I was always very nervous to go to graduate school because I didn't think I was good enough. You know, and a lot of people have imposter syndrome, and you know, everybody feels that way. Is a really important message also. At some point, that feeling is held by almost everybody. So just knowing that you're not alone in that feeling is really important.
Yeah, that's good advice. Thanks.
Yeah.
So what's coming up next for you? If you're thinking, you know, and maybe even idealistically, what do you hope is coming up next for you?
Yeah. So with the domestication work, I feel like every time we do something, it uncovers 1,000 new things to look at. So you know, getting more into the food science aspects of the volatile compounds and the sensory attributes and all that stuff is super exciting to me. So that's one place that we're moving into, is really trying to figure out, like, how are these things different based on the compounds that they produce and the smells and the taste that they produce. And then we're also, so Ezra and Kim we have this hypothesis that, you know, so miso fermentation, for example, there are many, many different bacteria and fungi and yeast that are involved in that fermentation process. And we want to know how they interact with each other. You know, how do we get this consistent product every time? How are they not dying off at certain phases? And it follows a very clear path of fermentation. And so we're trying to figure out, we'd really like to know some of the interactions between, how are they feeding off of each other, figuratively and literally, you know, how are they actually feeding off of each other? So that's another kind of big place where we're going now.
Well, I'll look forward to hearing about that. That sounds really interesting.
Thanks.
So last question, unless there's any other audience questions after this. What's your favorite thing about being a scientist?
Oh, by far my favorite thing is like discovering something new. There's nothing like that feeling. It's like scoring a goal or something. You know,
It's like finding a buffalo nickel.
Exactly. Thank you. Yeah, we're full circle now.
Yeah.
Yeah, there's nothing like discovering something that's never been discovered before. It's the coolest thing you can ever experience. And you know, I feel so lucky in this job that I get to just ask questions and try to answer them. And sometimes they don't get answered, but it's this. You get this rush from finding these very, very interesting things. Yeah.
Well, we're lucky to have you answering our questions tonight. We've got some good recommendations too for the sake distillery brewery and for the best metal detecting spots. So I think we're all taking home a lot. Anybody else want to ask any last questions of John? And otherwise we can wrap up and chat and eat some pizza. All right. Thanks so much. Please join me in.
Thank you.